In this video, I will be explaining about the structure of DNA. DNA is a nucleic acid, which is a polymer of nucleotides. Nucleotides are made up from the phosphate, a pentose sugar, and a nitrogenous base. In DNA, the pentose sugar is deoxyribose, and the bases are adenine, thymine, cytosine, and guanine. When you are asked to draw a nucleotide, this is the accepted format, with the circle being the phosphate, the pentagon being the sugar, and the rectangle being the base. This is the same whether it is a DNA nucleotide or an RNA nucleotide. All that changes is the name of the pentose sugar and one of the bases. As with most biological molecules, nucleic acids polymerize with a condensation reaction. This occurs between the phosphate and carbon-3 of the next nucleotide. This forms a strong covalent bond called a 3'-5' phosphodiester link, and this helps to form the sugar-phosphate backbone, which is an integral part of the DNA structure. DNA is a double helix. The chains are held together by hydrogen bonds between the bases. So you can see that you've got a sugar-phosphate backbone with the bases poking out to form almost like the rungs of a ladder. And what we find is that we have complementary bases. So adenine always pairs with thymine, and guanine always pairs with cytosine. The strands run anti-parallel, which means that one is in this direction and one is in that direction. And that is important when we come on to DNA replication later. The complementary nature of the base pairing means that one strand can act as a template for the other strand. Structurally, it is 2 nanometers in diameter, and you get a full turn of the helix for every 10 base pairs.